Hello, and welcome everyone to Season 4 of Contact's Produce Safety webinar series. In the last year of this webinar series and our current grant, we'll address some of the topics that have been in the news related to produce safety, as well as our research with emphasis on practical applications. This series is part of a larger industry outreach and risk mitigation project called Contact, or scientific challenges and cost-effective management of risks associated with implementation of produce safety regulations. This project is supported by a specialty crops research initiative from the USDA National Institute of Food and Agriculture. Any opinions, findings, conclusions, or recommendations expressed in this presentation are those of the speakers and do not necessarily reflect the view of the U.S. Department of Agriculture. Probably after about 25 webinars, you know who Contact is, but for those of you that are joining us for the first time, Contact is made up of several universities and government institutions. It's about 50 faculty, staff, students, and volunteers, not to mention the support of our Produce Advisory Committee, or PAC. It takes a village, and you're part of it. We're very appreciative of each individual's support and ongoing efforts to make Contact a grant champion by you all. Contact is now in its final year, so we are very grateful for your support as we're planning to submit a Contact 2.0. If there are any things you would like us to focus on, questions you would answered, resources you would developed, or if you would like to write a letter of support or serve on our Produce Advisory Committee, please reach out to myself or Dr. Michelle Daniluk at the University of Florida. I will drop our emails in the chat, but we would love to hear from you. I also want to make sure you're aware of our Contact website and YouTube page. These have all of our webinar recordings. We have key takeaway documents, more information about the grant, as well as some of those research updates, peer-reviewed papers, or different FAQ documents, and much more. You can subscribe to our page, at least on YouTube, and get instant notifications of when content is dropped. For instance, this webinar recording. Okay, why we are all here today. We are here to listen to our fabulous group of speakers, Laurel Dunn, Manan Sharma, and Keith Scheider. We have our Research to Reality segment, and this is biological soil amendments. All of this work is directly from the Contact grant and will be now shared with all of you. So while I let Laurel set up her screen, she will tell you a little bit about herself with her adorable background.
Thank you, Laura. Hi, everyone. Laurel Dunn. I'm at the University of Georgia, an associate professor and extension specialist, and I have had the pleasure of working with the Contact team over the past several years, particularly focused with the biological soil amendment group and biological soil amendments of animal origin in particular. So you're going to get to hear about some of the work we did with Keith and Manan in their presentations. And I'm going to just do a little bit of an overview on where we are, give people a refresher, a recap of the produce safety rule and some of those requirements, and really how our biological soil amendments are used, why they're used, and then discuss some ways that Contact also has engaged the produce industry throughout the course of this project. And hopefully, if we get another round of funding, we'll be able to continue with some of that engagement and collaboration. Knowing that most of us are familiar with the produce safety rule, but wanting to get people caught up just in case, the things to keep up with here are this part of FSMA covers everything really from growing, harvesting, packing, and holding. And particularly when we're discussing biological soil amendments, we're really going to be focused on this growing portion of it. Keeping in mind that this rule and really what we're doing in food safety in general is all about our risk reduction, our hazard reduction while we're producing fresh produce, not necessarily that risk elimination. Like, we would love it if we could eliminate all risks from our food supply. We're really focusing on these big areas, these big buckets that we can reduce a lot of our hazards that are just going to be inherent to produce production. So when we get thinking about our biological soil amendments of animal origin, so these are going to be just our refresher, our amendments that are going to be coming from animal products, right? So our fecal material, our bone meal, we'll get into that on the next slide. The reason we're using a lot of these is to promote really our soil fertility, so adding nutrients. We think of our NPK values, our nitrogen, phosphorus, potassium, micro and macronutrients that we're adding to the soil to promote plant growth, as well as just soil conditioning in general. We have commercial formulations that are available, primarily treated formulations that you can be commercially purchasing, as well as a lot of these materials just are waste material from animal agriculture. So again, that fecal material, those animal mortalities, bones, blood, things like that are left over from animal agriculture, but have another usage for soil amending. They can be a money-saving feature or have money-saving characteristics over some of our synthetics. This is going to depend on where you're located. About a quarter of our farms that are using biological soil amendments really try to source pretty locally, just because you're transporting tons of this material. It's obviously going to be more expensive. So can be a money-saving feature, and also can really help a lot of our organic farms that might be limited in the synthetics they can use or not able to use synthetic fertilizers. They still have this source for nutrients. Then we have our breakdown into our treated and our untreated biological soil amendments of animal origin, or BSAAOs. So when we're saying treated, these are going to have been treated in some manner to reduce our foodborne pathogenic microorganisms, or at least those are the ones we're obviously concerned about from a foodborne safety, a food safety perspective. So when we look at some of our common amendments, something like fish emulsion is typically heated up pretty hot to reduce a lot of pathogens, and so you get a longer shelf life of that material just by the way it's made. Our bone and feather meal, the same way. So like our leftover products, again, from animal agriculture, these are going to be heated up or steamed before then they're ground into these meals that can be applied. And then a lot of us are probably more at the home scale familiar with compost. If you have a backyard compost that you're using, scraps from the kitchen, where we're piling our organic material, different mixtures of organic material. And as those microorganisms are breaking that material down, the pile is getting heated up, and that heat is then killing microorganisms. These processes have that added benefit. Obviously, we're food scientists. We care about the foodborne pathogens being destroyed. But you also have some added benefits of things like our plant pathogens are typically being reduced by a lot of these processes, weed seeds, things like that. With these materials, our foodborne pathogen risk is going to be low. We never say zero, but it's going to be lower than, say, if we use something like an untreated biological soil amendment. So if we're using something like a cow manure or a pig slurry or some sort of manure from an animal production farm. In Georgia, I have to talk about poultry litter, particularly broiler litter, when they clear the birds out of the house. There's all that material that's left over, so bedding, which could be sawdust or peanut hulls or things like that. And then fecal material, dead birds that are left over, feces, things like that are left over in the house and are still a really good nutrient nitrogen source. Since these aren't treated, these could be a higher risk for some of our foodborne pathogens. So what we know as food safety people is just that our biological soil amendments of animal origin do often contain some foodborne pathogens. So things like Salmonella, E. coli. So it gets us thinking a lot of times, or getting a little defensive of like, all right, we know there's Salmonella in these materials. Why are we adding this in fresh produce fields if there's this likelihood for produce contamination? What value do our BSAAOs, particular untreated ones, have for fresh produce production? And so I just wanted to come up with some key points of why we still see it in fresh produce production and why it still has value, even if we can't treat everything that we use. Why are farms still turning to some of these untreated biological soil amendments? So I just have this graphic here, and obviously I have steps missing, so don't judge me too hard on this graphic. But what we have is a chicken farm that we're starting with at the front end. Chickens are removed from the house. And so you might have a skid steer, front loader, something come in, scrape that house clean. And then if their goal with this is to make a commercially composted poultry litter product for agricultural production or produce production, that skid steer is going to have to transport it to a truck. There might be another piece of equipment involved in loading it, or it might just dump it into these compost piles. They're going to have to be turned regularly, depending on the process that's being used for this composting. So probably more pieces of equipment involved there. It's going to have to be loaded up again onto another truck. And then when it's taken to the farm it's being delivered to, potentially unloaded, put on a spreader, and then finally transported to my production field. So a lot of equipment, a lot of time, a lot of handling to get this commercial composted poultry litter product. If we have a situation where I might be a mixed commodity farm, I might have chickens and other commodities that I'm producing. So I'm pulling this from my own farm or from my next door neighbor's farm. This is a little simpler process where I might scrape my chicken house out, again, stack it into these piles, be composting, hopefully using my validated process, using my thermometers with that. And then once that compost is finished and cured, loading it up again, loading it into my spreader and applying it to my field. So by maybe composting at home, I may have reduced some of these steps and may have really reduced some of my costs associated with this material. Then if, you're in an area of heavy poultry production, which we see particularly in Georgia, particular regions in Georgia will have a lot of this, and I know other areas of the country where you have neighbors or you yourself have chicken farms. It's really simple to, once the birds are gone, scrape the house out with the skid steer, maybe stack it up, or maybe immediately stick it into your spreader once it's dried out enough and immediately apply it to your fields. What you've done here is you might not have that treatment step, but you are saving a lot of time with the handling. You are saving a lot of labor. You're saving a lot of fuel costs. If you remember from that first diagram, there were a lot of vehicles there that were needing a lot of diesel to run those. You're reducing wear and tear on your equipment. You're really just speeding up this process a lot. A farm who's really doing a cost or a time analysis in this might be very driven to select some of these untreated biological soil amendments just for ease. Then if we also look at these from the standpoint of nutrient retention. So we have our nice little nitrogen cycle here. Hopefully you're not having nightmares or flashbacks to your environmental science classes. We'll really start in the middle of this graph with our little bunny here who might be depositing feces into the soil. And we know what's happening with that. So that would be like us applying our untreated biological soil amendment to the soil where that rabbit is. And we know that once it's applied, we're starting to get that decomposition. We have microorganisms in the soil that are further breaking our nitrogen-containing compounds down into things like nitrites, which are then getting converted to our nitrates, which our plants are really happy to take up. And then we have our denitrifying bacteria that might be further converting some of those nitrogen-containing compounds to atmospheric nitrogen. So we have a lot going on in this system for our compound that might start as our animal feces or animal product before it's actually converted into a usable form for our plants. But there are a lot of nitrogen-containing compounds that are being created through this process. Now, unfortunately, what's happening is when we're composting and we stack our litter into this compost pile, we are heating it up. We are reducing a lot of our pathogens and weed seeds and things like that in the pile. But we also have a lot of total nitrogen escaping from the pile throughout the process of this composting. So again, we have that ammonium going to nitrite, and then hopefully finally nitrate, which again, our plants are really happy about. But we're also losing, again, that atmospheric nitrogen, or we're losing ammonia and losing that nitrogen that's bound in those compounds as it's leaving the pile. And nutrient loss is really a big concern for composting and for composted products in general. So while it's really good material and is really good for conditioning soil, there have been a substantial amount of studies. And if you really look at this one, you can see this is an old study, but I like this one because I really like their numbers. And it really seems to agree with the past 20, 30 years of research on nutrient loss in compost. So this group was examining cow manure that was being composted, and they found nitrogen losses, total nitrogen losses in their compost piles, ranging from anywhere to 19 to 40%. Carbon losses were about 50%. And then they found when there were significant rainfall events, they'd be losing things like sodium from their piles as well. So for someone like an organic producer who doesn't have the option of maybe being able to amend with synthetics throughout the season as easily as a conventional grower, this could be a really big concern with that total nutrient loss going into a production season. And then obviously we're concerned about it from a waste management standpoint. And these are really the big three things I picked out, but there are other reasons why we might be considering usage of this material. Again, I'm focusing on poultry because we grow a few chickens in Georgia. Overall, the U.S. poultry industry generates 10.2 million tons of poultry litter. Georgia is responsible for about 20% of that material. If you have a significant amount of material coming from one segment of animal agriculture, you're going to have to find some way to manage it. And if there's not a strategic waste management plan that might involve crop production, you can have issues with over-application to land, which can obviously result in eutrophication of waterways, where you're just getting crazy algal growth and really throwing off those ecosystems. can also get it if you have heavy loads of nitrites in that material, you can get that material actually leaching into groundwater. And so really being strategic with how we're managing this waste is crucial to make sure that we're not all swimming in poultry litter or cow manure or really a lot of these byproducts, these waste products from animal agriculture. So coming back to all of this, we still have to manage these safely. So if we're going to use them, we really need to understand that, yeah, these do have benefits for production, but obviously there are risks involved with it, and we don't need to be blind to those, and we need to be strategic with how we're using this material. So this can include things like preferentially applying things to our lower risk crops, which we teach pretty much in all the PSA grower trainings we have. If you have things like your agronomic crops, if you're growing a lot of cotton, a lot of grain crops or oilseed crops or things like that, try to preferentially apply to those, or crops that aren't coming in direct contact with the amendment, with the ground itself. Avoiding our contact with the edible portion when we're applying this. So trying to be careful when we're applying it, that it's going where we want it to go and that we're not getting a lot of wind drift or things like that when we're applying. And then we can't have a food safety talk without talking about record keeping, really keeping up with our material type, where it came from, storage, application date becomes a big one, particularly if we're using something like the National Organic Program 90 to 120 day application to harvest interval, which right now is the guideline that we're going by for the produce safety rule. But our organic growers, who are ones who are most likely to be using our biological soil amendments of animal origin, are probably relying on them the most. If they have that organic certification, fortunately, a lot of them are already really familiar with that 90 to 120 day application interval. And of course, that's been 90 days for our crops that aren't likely to come in direct contact with the soil or with that amendment itself. And then 120 days for those that are likely to be grown directly in that. The produce safety rule also, we have the requirements of you must make sure that you're handling these and you're storing these in a manner that you're not going to be getting cross-contamination to other things in your operation. So obviously we're very concerned about the produce, but also water sources, our food contact surfaces, really trying to make sure that they're stored and they're handled in a way to keep other things from getting contaminated. And as always, when we're applying, we must avoid that direct contact, particularly with an untreated biological soil amendment, must make sure that we're not contacting the crop or the edible portion itself. So as a part of contact, one thing we did was we did survey growers just on some of the perspectives about biological soil amendment use. And this was a poster you may have seen at IA. We did this towards the end of last year, beginning of this year. So I guess about a 4-month period, and we're surveying growers. And so we found is about half of our growers do have concerns, particularly concerns about the safety of biological soil amendment use. They did express that they do understand that there is a hazard here, and this is a hazard that likely needs to be managed and that they need to be aware of. Ours, I like to think that our 50% in that neither concerned nor unconcerned, and then the unconcerned ones maybe are less concerned about it, just from the standpoint of maybe they're relying more on synthetics and they're using less of these biological soil amendments of animal origin. And we also asked them, if you are treating a biological soil amendment of animal origin on your farm, are you keeping good records and are you monitoring? And what we found with this was that no one replied yes to record keeping and monitoring. 67% of our respondents, now keep in mind, we had 9 respondents for this one, so maybe not the most impactful we're not able to draw huge conclusions here. But 67% of them said that they weren't doing any record keeping or monitoring. And 22% of them were doing some monitoring, but they weren't keeping any records to indicate that maybe they had safely processed something and were treating things as they should have. So in summary, currently both treated and untreated biological soil amendments of animal origin are permitted and are frequently used for fresh produce production. They're really a valuable input from a fertility and a soil health standpoint. But we still need to make sure that care is being taken, particularly for using these untreated ones, so that we're really reducing the likelihood of produce contamination from those foodborne pathogens that we know can be present in the material. There is research that is currently being conducted to assess both the survival and the transfer of some of these pathogens from our BSAAOs to fresh produce. And with that, I am going to stop sharing and hand it over to Dr. Scheider.
Hey there, everyone. I'm Keith Scheider. I'm in the Department of Food Science and Human Nutrition here at the University of Florida. am a food safety microbiologist focusing on produce. I'm going to go over some research that we've been doing over the last couple of years with Contact and some grants before that, and just go over what we've seen here in Florida and in partnership with some of the other universities on this grant. So, as an overview, we examined the survival of pathogens, Salmonella and E. coli in microcosms here in the university. We've looked at the survival of E. coli in the wild in field trial with generic E. coli. Then we've looked at the effect of BSAAOs on the survival of E. coli that was inoculated into the soils. So we'll talk about these each in order. Laurel's already given you a great definition and history of biological soil amendments. Concern to us was looking at the NOP's 90 and 120 day standard for entry into the field and whether or not pathogens that would make into the field operations, how long would they last and what was the effect of these soil amendments on those particular pathogens. But let's go back and look at some microcosm studies first. And this is just naturally occurring E. coli that we isolated from dairy manure that was used here in Central Florida. We looked at two temperatures in microcosms. These were technically mesocosms, 1 kilogram mesocosms, called at 20 degrees and 30 degrees C over time. And they were taken from two different areas here in Florida, Live Oak and Citra. Live Oak has a slightly more sandy soil, where Citra has a slightly higher loam content, but they're still considered sandy loams. As you can see from this, the difference in the location really doesn't matter a whole lot, or didn't matter a whole lot. What we did see, though, is that the holding temperatures made a huge difference. And we see this sort of die off in the first 60 days. And then we hit a kind of persistence phase where we still had platable cultures out to about 180 days at the 20 degrees. So as the temperature went up, the faster the decline. And that's a trend that we've seen through most studies and through most of the literature. This particular study, what we were looking for when we were looking for endpoints were two negative enrichments. So what we did when we no longer could plate, we would just do an enrichment of the sample. And again, if it was positive, we just marked it as positive until we got two negative enrichments. Didn't even call it negative. Crazy thing was in these microcosms, we were able to isolate organisms, at least had positives, out to 420 days. So long, in fact, that the student graduated before he got done with the project, and some of the incoming students had to take over. So even though the vast majority of these numbers dropped dramatically early on and a significant effect due to the temperature, the persisters remained in both sets of samples. This was kind of piqued our interest a little bit. Another study that was done was the effects of moisture over time. And again, this is just a kind of a little snapshot. We also did sandy soils that didn't have as much water holding capacity, but in these sandy loams, we were able to saturate a wilting concentration of a 50% moisture content all the way up to 35%. And as you can see, the prevailing thought would be that the more moisture, the happier they would be. And we saw the opposite. And a lot of the differences here are not significant, though we do see a trend that the lower moistures tend to have higher survival. And this is again at 20 degrees C, and again, we're looking at E. coli and Salmonella this time. And again, thinking about, are the organisms as effective, or are we maybe affecting the predators within the soil microbiome that would affect the other microorganisms? So we looked at this, but we did see that higher moisture really led to greater survival. And that's going to come back up when we do the field trials, because again, in the literature, and you'll hear from Manan later, again, rainfall sometimes correlates with a bounce back, but some of the stuff that we've seen, especially in the sandier soils, the organisms really don't care about moisture too much. So we went to field trials, and this is in a research center up in Live Oak, Florida. These plots were roughly 3 meters by 10 meters. And they were amended with untreated bovine feces that came from the dairy. And we tracked the naturally occurring E. coli, which was present in the manure. And then we monitored these for approximately 300 days. This work was done by Samantha Chuckich. And as you can see, we're still seeing that rapid die-off here. And this is the fall season, and we're getting fairly rapid die-off here. both in core samples and surface samples. And we're really looking whether or not, in this case, solar radiations and the higher fluctuations in temperature at the surface really made a difference. And it really didn't. We were getting reduction right around 28 days. We were getting a rapid reduction. And then again, we had the persisters where they'd go in and out of platability, but we still were able to, again, isolate or get those persisters to come back in an enrichment all the way out, way past 250 days. There are three different types of amendments here: the control and the green line at the bottom there. On two occasions, we did have little spikes after rain events where we did have runoff. So there occasionally we would have runoff from the control plots. But for the most part, again, we get this rapid decline right about 28 days in the fall when it was fairly cool. But we were able to, again, get recovery all the way out to post 250 days here. Now, we repeated this study in the summer, and you notice, again, the same thing with the microcosms. The warmer temperatures, the faster the die-off. And again, the rapid decline that we saw occurred around 28 days, and we stopped getting positives somewhere between 100 and 150 days. So again, much faster die-off. Both sets of graphs here, we're looking at a light, medium, and heavy application of manure to the field, which is medium was 1x, light was half the agronomic rate, and heavy was a 2x application of the manure. But yeah, you can see again, the warmer temperatures did lead to faster die-off. Moving on, this is another study. This is work done by another student, Manan Baker, and we were trying to assess here, again, in microcosms, the effect of the soil microbiome on their survival. So what we did, and in this case, we were working with coli 157 variants. We have STX -1 for 1 and 2, and STX 2 positive, and then a 1-2 positive. And what we noticed is that the strain really didn't matter too much. And we have two clusters here where we took the soil and we autoclaved it 3 times for 90 minutes each, trying to kill off as much of the indigenous microorganisms as possible. Interestingly, we got most of the vegetative cells, but there was still a large number of spore formers left in the soil. We tracked the metagenomics of all this, but it's not going to present it here. But you can clearly show the two clumps here that when the soil was left in its natural state with all the other microbiome intact, the organisms died off much more quickly. The E. coli died off much more quickly. When we autoclaved the soil and we removed most of those vegetative cells, that die-off occurred much slower. And again, regardless of whether it was any of the 3 different strains that we used, it clearly showed that there was a difference between the two. And there were subsequent experiments trying to isolate potentially what the antagonistic organisms were. And again, we were looking at potentially nematodes and other gram-positive and gram-negatives. Never were able to find a single organism that was responsible, but it was clear that the microbiome does have some effect, beneficial effect, as again, we've seen in the literature that the more complex microbiome, again, aids in the reduction of these microorganisms. We'll get back to that later. And this is for E. coli. Now the student's work, this is Ellen Gutierrez. And we were looking at Salmonella, and these two students were going in my lab at the same time, and we expected to see the same thing. Crazy thing that is we didn't. So what we did is to get rid of all the spore-forming and heat-resistant cells, we irradiated the soil to basic commercial sterility. And we applied Salmonella to see if, in fact, again, Salmonella behaved the same way as E. coli. So we have irradiated, untreated poultry litter, and then just the untreated versus the irradiated. And we expected to see that the natural, when treated with the diverse microbiome, increased the die-off, or we had the same effect that we saw for E. coli. We didn't. And after much consternation and puzzling that's out and work by the student, we found out that Salmonella was really being affected not by predators or any other of the microbiome organism in the soil, but rather the ammonification, I believe is what Laurel said in that one diagram, but the ammonia production. And we would take the soil and we would remove the microorganisms that were responsible for breaking down urea. And when we added them back, or we would add, and we tracked the ammonia production, Salmonella was really affected by the ammonia production. And again, that really aided in the removal of Salmonella. So in this case, Salmonella behaved a lot differently than we saw with the E. coli. So the behavior of these particular pathogens, which we thought would be similar, was not. Matter of fact, Salmonella really didn't care about the microbiota too much, whereas E. coli was very sensitive to what was left in the soil. So that leads us to the current study that we ran as part of the contact grant. And again, this was done in Live Oak, Florida. The plots are slightly smaller this time. We ran 1 by 3 meter plots. As you can see, this little flyover here, we would inoculate with E. coli, and then we would track the survival with the amendments that I'll discuss in just a second. Now, this was work that was done by Charles Bency Apollon and input from 2 really wonderful postdocs that I had, Cameron Bardsley and Brenda Carell. Just want to give them a little bit of credit. And this is what we saw. What we did was we did E. coli onto the soil untreated. And again, remember, this is a sandy north central Florida soil. We added a fully composted poultry litter. And then we added a heat-treated poultry pellet, which was that litter process that Laurel was talking about, except it's treated to inactivate all the microorganisms and then pelletized. So all the organic benefit, none of the potentially harmful microorganisms associated with it. And what we saw was over this 140-day trial, and that corresponded to the growing season of onions, which were grown as a separate experiment as part of this whole study, that when we added the poultry pellets, it actually increased the survivability. Now, we were able to get survivors or persisters all the way out towards the end of this experiment, but we had platable organisms, which is, again, phenomenal, actually, all the way out to 140 days. So we had, again, that rapid die-off that we've seen in all the studies up to this point, except the addition of just literally less than a kilogram of amendment to that 1 by 3 meter plot was enough to affect the survival of the E. coli. This was a generic E. coli that was applied to the field throughout the course of the experiment. Now, the poultry litter also had an extended survival compared to the control, though not as much as the heat-treated poultry pellets. If you look at the control, which is that orange line, the E. coli on the sandy soil died off the quickest or became unrecoverable or the quickest. So what we're seeing here, at least what we're postulating, is that the addition of organics aids in the survival of the E. coli, but the presence of the more diverse microbiome that's in the unheat-treated poultry litter had a beneficial effect. So we were getting this survival benefit from the amendment, but we're also getting the benefit of the diverse microbiome. So the composted litter fell right in the middle. This is one of those things where the poultry pellets are, in the beginning, a safer option because, again, we've heat treated them and there's no chance of improperly composted manure introducing pathogens to the field. The downside is that if the field becomes contaminated after the application, now we have the negative of that aiding in the persistence. So darned if you do, darned if you don't. So we wanted to look at this. And the interesting thing, again, is that we were able to get platable numbers out to 140 days. And quite honestly, it went out to almost 156 days. Because as part of the onion study, which I'm not going to present here, we were able to track it through curing in the soil. And quite honestly, we were able to actually pick it up on the onions themselves. So again, happens to be very timely. The results are showing that, again, they can survive for a long period of time, then actually transfer to crops grown in the field. Now, one of the things about this and what a lot of farmers will do is they will come in with a biological soil amendment pre-planting as they're prepping the field, and then they'll use synthetic fertilizers for all subsequent fertigation throughout the growing season. And we wanted to ascertain what effect the application of a synthetic fertilizer would do. And that's shown here. And here we looked at the control, poultry litter, the heat-treated poultry pellets, and then the addition of synthetic by itself, synthetic plus the poultry litter, and then synthetic plus the heat-treated poultry pellets. And they cluster again. The synthetic did not enhance the survival. Matter of fact, the synthetic amended soils along with the biological amendments actually again, had lower counts. And these are not significantly different. There was a lot of variation here. But what we did see, again, the trend was that the synthetic wasn't adding any survival benefit and may, in fact, actually lower the survival profile of E. coli in the soils. And again, this was all done in microcosm, where the previous study was done actually in field. A couple of take-home points here. One, we know from surveys that we've done that biological soil amendments can be found in pathogens. The applications of untreated or raw manures can introduce pathogens to the field. As Laurel pointed out, though, properly composting or treating them can eliminate these pathogens quite effectively. One thing we do know is that we do get this rapid die-off, but these organisms can persist for extended periods of time. Again, in microcosms, we saw extended out to 420 days. For the grant where we did side-by-side with our onion trials, we got survival up to about 140 days. And beyond that, the presence of amendments actually aided whether it be the carbon, total organic carbon in there, the other commensal organisms. The presence of that on the soil helped the survival of E. coli compared to unamended soils. We did see, though, that the unheat treated poultry litter did fall in between lack of amendment and the heat treated poultry pellets, showing that the microbiota did aid in the die-off of E. coli, which is, again, calls back to, again, that microbiota being, again, maybe predatory to the E. coli. One thing we also want to point out is that the lab studies differ sometimes quite substantially from the field studies. Now, remember, one thing that I haven't mentioned yet is a lot in these field trials, what we're doing is we're not only measuring the survival, but we're also looking at weather conditions as well. These include irrigation, rainfall, solar radiation, air temperature, soil temperature, wind, and again, none of the ones that we tracked correlated with die-off other than the temperature, which again shows what the microcosm showed, that we had much more rapid decline in the 30 degrees compared to 20 degrees. But other than that, all the other parameters that we saw didn't have a strong correlation. So again, that is different to what we see in some of the literature. And I think Manan is going to be addressing that in the next presentation. But the big question, again, is the levels that we're seeing, these persisters and these very low levels, do they pose a risk? And that's a hard question to answer. We're seeing, again, survival, while albeit very low numbers, we are able to isolate them out of the soil for an extended period of time. So with that, I'm going to turn it over to Manan and let him wrap things up.
Thanks so much, Keith. That was a really good talk. And while I'm sharing my slides, I'll tell you a little bit about myself. My name is Manan Sharma. I'm a research microbiologist at the USDA Agricultural Research Center here in Beltsville, Maryland. And yeah, I work in the Environmental Microbial and Food Safety Laboratory. So I said I'm a research microbiologist here in the Environmental Microbial and Food Safety Laboratory. The work I'm going to present today is mainly done by a postdoctoral research associate here, Ray Jiang, who's part of the Contact Project. I really appreciate his hard work and analysis in this project, but was also contributed to by Ellen Gabriel, who's a graduate student now at Virginia Tech, Dr. Alan Gutierrez, who's a postdoctoral research associate here, Cheryl East, who is a longtime microbiologist here at USDA. So I appreciate all their contributions. And we're going to talk about side dressing today. So Keith and Laurel both gave fantastic backgrounds and primers for what I'm going to talk about here today. And if you don't know, I'm sure a lot of you do, side dressing is a common agricultural practice, and basically to provide nutrients during crop growth. And Laurel and Keith both laid out a lot of advantages to using organic fertilizers in soils, but some plants need a little more nitrogen, need a little more nutrients after they've emerged during the growing process. You can see here on the left, this is Payman Aminabadi, who was a contact collaborator at UC Davis, manually applying some organic fertilizers to romaine lettuce heads out in the field. And you can see there's different ways to do this. On the right, this is a tractor with a manure injector, side dressing a bunch of corn, getting ready for harvest in Minnesota. So there's a lot of different ways to do this, but the idea is to supply nutrients to these crops after the plants have emerged, as opposed to providing the nutrients to the soil before planting. And just a little bit of background, I think some of Keith's graphs really showed this well. better than my little side panel of dots. I know it's late in the day, so sorry to geek this out with you. But one of the reasons we were interested in side dressings is one, we know that a lot of organic leafy green growers use organic fertilizers, so biological soil amendments, sometimes of animal origin, to side dress leafy greens when they're producing them organically. And some of those soil amendments really haven't been evaluated for their effect or ability to support pathogen survival in soils. Keith talked a lot about heat-treated poultry pellets. We'll talk about that too, and a couple others. So we really wanted to evaluate those amendments, because we thought they were novel and emerging to be used in leafy green production environments. And one of the things that Keith showed, and hopefully what in this group of 6 graphs on the right-hand side, is that the pathogens or the E. coli in manure or in soils that have these amendments, it doesn't always decline right away. There are some times where the population stays steady for a few days and then starts to decline. There are some cases where the population actually goes up by a log or 2 and then comes back down. So if you're side dressing and applying those soil amendments into the field, do you see this kind of effect? So if you do have some external contamination, are those pathogenic cells going to be able to actually grow or basically not decline? And will that lead to increased likelihood of contaminating the crop? In this case, romaine lettuce in our experiment. So that's really what we're trying to investigate here. And the biological soil amendments that we evaluated in these experiments, heat-treated poultry pellets, which Keith talked about, and you can see our NPK values. These are the NPK values listed on the package or listed with the actual amendment. We did test these two, and they come out slightly different. But heat-treated poultry pellets, I don't think we need to talk about that anymore. The seabird guano was something that Michelle J. Russell, who is a contact collaborator at UC Davis, told us that leafy green growers are using in California in organic production. And again, just keep referring to organic production. Organic growers can't use chemical fertilizers for the side dressing. It does have to be organic. So this is a really good high nitrogen fertilizer to use. So seabird guano. And then corn steep liquor, which is a byproduct of wet milling corn, stays at a high temperature for 12 hours or more, and then comes out, has some nitrogen in it, could be used. Laurel used the term biological soil amendments of animal origin, which is the common term. I'm just going to use biological soil amendments. And the difference in the terminology is that this corn steep liquor is not of animal origin. So that's why we stopped at biological soil amendments. And so if you're wondering why we're inconsistent on terminology, that is why. So just to geek it out a little bit more on your Wednesday afternoon. So as Keith mentioned, what we're worried about here is an external contamination event to the field. So the way I like to think about this, the way we went about approaching this experiment is, say that you're using surface water. For some reason, you might not be treating it or may be treated inappropriately. We know that surface irrigation water can contain bacterial pathogens. Okay, so you're irrigating your crops, you put down your side dressing into the soil, your plant has emerged from the soil. Say that water gets in your soil with those amendments. Can any of these pathogens or organisms grow or not decline in that soil and then transfer to the edible portion of the crop and then result in human illness? So this is a scenario that we think about a lot, and this is why we wanted to investigate this. There are other ways that you can get external contamination of the soil, not just directly from the soil amendment. And it should mention all of these things are treated soil amendments, so they have been through some heat or composting or validated process, so they are not starting out with a pathogen load in them. So again, our objective was to evaluate the longitudinal survival of a non-pathogenic E. coli, an E. coli 0157H7, which is a pathogen, and then a Salmonella infantis strain, co-inoculated in these soils that are side dressed with organic fertilizers or the biological soil amendments that I mentioned, and then evaluate their transfer to romaine lettuce. And as I mentioned, this work was conducted by Ray Jones. You can see him on the right there. All right, so we had 2 different studies in this work. So study number 1, we co-inoculated the soil with E. coli 0157H7. These are rep strains as classified by the Centers for Disease Control and Prevention. They're chlorphenicol resistant. They're isolated from lettuce outbreaks. The CDC classifies these as rep strains because they're closely related by their view. And so we obtained these outbreak strains from them. We also use the non-pathogenic strain TBS 353. This is a really common commonly used E. coli strain. In fact, in other objectives of the contact project, we've used this strain to show continuity for different objectives. That helps out with the modeling and the consistency. This strain was isolated by Trevor Sesso's laboratory when he was at UC Davis. It's become a really good surrogate. It's showed a lot of environmental hardiness. It's rifampicin resistant. And so we used it here. So we co-inoculated those 3 strains together, 2 0157 strains and the TVS 353 strain. In study number 2, which was done after study number 1, we inoculated the soil with Salmonella infantis. And then this is a Salmonella infantis strain isolated from surface water in the mid-Atlantic region. It's actually multi-drug resistant. So we're able to trap it pretty easily. And our lab isolated this working on the CONSERVE study, which was another USDA NEPA project. Okay, so side dressing, our side dressing application scheme. Okay, so we actually had 2 side dressing events. And we had a scheme here. And this is based on what people are doing in California. But in our treatment scheme, HTPP2 means that you had 2 side dressing events, both with heat-treated poultry pellets, or HTPP. HTPPCSL means the first side dressing treatment with the poultry pellets, and then the second side dressing treatment with a corn steep liquor. Seabird guano 2, the same thing, 2 side dressing events, both with seabird guano, the CBG CSL, first treatment, the first side dressing, seabird guano, second side dressing, corn steep liquor. We had an unamended control, which means the soil was inoculated, but no amendment was applied. And we had a negative control as well, just to look for cross-contamination. So our first side dressing event occurred 11 days after romaine lettuce plants were transplanted into the soil. We actually grew the romaine lettuce as seedlings and then transplanted them into the planters that we use. And the second side dressing event occurred 21 days after romaine lettuce transplanting. And then after that, that second side dressing event was when our soils were inoculated with E. coli or Salmonella. And then we analyzed those soils up to 28 days. And we harvested the romaine lettuce on day 28 or day 21. So our romaine lettuce variety was an organic variety called Coastal Star. And after we had applied the second side dressing event, we have 2 really nice biosafety level 2 growth chambers in our lab in Beltsville. So we were able to grow our lettuce there while the soil was inoculated with either E. coli or Salmonella. We grew these with 10 hours of daylight at a constant temperature of 20 degrees Celsius and a relative humidity of 60%. Going through the soil side dressing treatments before, as I mentioned, the soil was inoculated with either non-pathogenic E. coli or 0157 E. coli, or in the second study, Salmonella infantis. In both cases, the lettuce was irrigated every 2 to 3 days with a relatively consistent amount of water, 400 to 800 milliliters. And then study 1, with the romaine lettuce was harvested at 28 days after the second side dressing event. And in study 2, the romaine lettuce was harvested 21 days after the second side dressing event. But we tracked the Salmonella to day 27. All right, so I'm going to show you guys a bunch of graphs and tables right now. Don't want to overwhelm you late in the day. Just please look at the trends. We're not looking at specific data points here, but we're always happy to answer questions about this. But if you look at our E. coli TVS 353 survival and amended soils, again, this is after the second side dressing event, going out 28 days. This is just survival in the soil. You can see all those lines running together, all winding up basically within like between 0 and 1.5 logs. Some error bars there. Pretty consistent. Not what was unexpected is you see that magenta line right here. That's the unamended treatment. And as Keith showed, we expect to see faster declines, lower levels of survival in the unamended treatments. We didn't really see that with the TVS 353. So that was interesting that we did not see that. This is the longitudinal survival of E. coli only 7, again, in the side dress soils. A little more space here, a little more diversity, a little more variety in the declines that you observe here. And again, we see that unamended soil not really supporting those faster declines at lower levels. You see it actually doing pretty well, supporting a higher level relatively to all those others. Ray modeled these trends, and with the TVS 353, he came up with a log linear decline. And modeling scares some people, get complicated. But in this case, the models are really good to illustrate some of the trends. And it shows you, like with the TVS 353, those are pretty consistent declines. Those are consistent, kind of steady declines, pretty even. And maybe you anticipate that because our environmental conditions are pretty planned out, pretty consistent. But with the 0157 on the right-hand side, you see a variety of models fitting the data, log linear models, again, that consistent sort of decline. And then you see some other biphasic and Weibull models, which basically indicates that there's different phases of declines. And in this case, you have a very quick decline at the beginning between days like 0 and 14, and then it slows down and comes back. But it's interesting, like these are all, everything is declining. We don't see a lot of survival. We don't see a lot of areas where it's not declining. We see it either declining or I'd say relatively quickly. But that was interesting with the unamended soils, but you can see they're all pretty much winding up in the same place. And that's interesting. Now, if we look at the data for the transfer of the coli if we look at the declines, apologize, we wanted to measure the declines a couple of different ways. We wanted to measure the actual amount of the declines in a certain period of time. So we chose 21 days. And we also wanted to measure how long it took to get a 3-log reduction. So I'm going to focus on the time reductions here because a lot of the biological soil amendment rules are based on time. And so if you look at the TBS 353 data here, you see these declines. The amount of time it takes to get a 3-log reduction is basically between 17 days and 20 days. And if you remember from the graphs, those are all the graphs that had those straight lines just going down, right? So those are pretty tightly bunched, pretty decent. But if you look in the red circle now, so now if we're looking at the 0157H7 declines and those side dressing treatments, you see a lot more variety from 13 to 22 days to get a 3-log reduction. And like the unamended being on the higher end of that, again, which was pretty interesting to us, not really expecting that. And you see the heat-treated poultry pellets with the double heat-treated poultry pellet side dressing HPP2 also having pretty high value here. So I think those are some interesting things. Again, you're evaluating them under like very consistent environmental conditions. And now when we look at the transfer of E. coli from side dress soils to romaine lettuce, which was done on day 28, you can see we've got about 13% of our TBS, we're covering 13, our samples are positive for TBS 353. 11% of our samples are positive for 0157H7. I should note that there was no quantification. All of our E. coli, we tried to quantify these when they were positive. All of our lettuce samples were below 0.56 log CFU per gram of lettuce. So this is a really low number that's transferring. But we break this out a little bit further and look at which type of tissue sample that we're looking at in the romaine lettuce, and we break it down by side dressing treatment. If you look at the third column, whole lettuce head samples, so 3 whole lettuce head samples are sampled for each side dressing treatment. And then we took inner leaf samples and then outer leaf samples. And what the positives are in red, which is a lot of whole head lettuce samples, and a lot of outer leaf samples are positive here. Very few inner leaf samples, which is to be expected, right? Because the outer leaves are the ones contacting the soil. And again, this is a really low level of contamination. Okay, so if we move on to our Salmonella infantis strain here, like I said, this is evaluating the survivals in the side dress soils, the same regimens that we used previously. And what you see here now is a little bit of a difference between how E. coli 0157H7 and TBS 353 survived and the Salmonella infantis survived, because you can see this again, this magenta line here. This is the Salmonella in the unamended soils. And this looks a little more like what I think Keith observed, where with the unamended soils supporting a more rapid decline and survival at lower levels, where those when the side dress soils with the biological soil amendments, seeing survival at a little bit higher level. So this is interesting that the Salmonella and the Salmonella infantis and the E. coli did different things. When we model this out, again, just to help us observe the trends, we see the unamended side dress soils, the soils that received no side dressing, declining much more quickly, the Salmonella declining more quickly in them, and then the other ones declining a little more consistently, a little more slowly compared to the unamended soils here. And so biphasic and log linear models here showing us the patterns. And so I thought this was really interesting that the Salmonella infantis, which was an environmental isolate, really declining much more quickly in our soils. And again, if we plot this out looking at the the amount of time it takes to get a 3-log reduction based on the side dressing treatment, you can see the unamended down here, you get a 3-log reduction in about 6 days. Whereas with the double heat-treated poultry pellet side dressing treatment, it's taking close to 20 days to get that reduction. And again, with the seabird guano double side treatment, you get about 20 days to get that 3 log reduction here. I should admit, we should have mentioned this before, and it's kind of a weird place to bring it up now, but our soil comes from our farm in Beltsville. We do have a 7,000 acre research farm here, and it is a sandy loam soil that we grew these lettuce, and that's what we amended it in. So now if we look at the transfer of Salmonella to our romaine lettuce heads, we grouped these a little bit differently. We grouped the side dressing treatments, the similar treatments, the heat-treated poultry pellets and the seabird guano here, because they both received the same treatment. So we grouped these together, and then we grouped the corn-steep liquor or second side treatment together, if that makes sense, and then the unamended. And you see a lot more positives here. The positives again in red. You see a lot more positives here for the Salmonella than we did for the coli 0157 or the TVS 353. And I think this is probably a function of us harvesting earlier on day 21 versus day 28. The reason we harvested on day 21 is, no joke, there was a government shutdown that was expected. And we thought that the government was actually going to shut down. And we had put so much time into these experiments. We definitely wanted to go through with the harvest and the analysis. So we actually had to move this up to day 21. But we were able to track the survival in the soils to day 27. So yeah, government shutdowns have consequences, but we did manage to get this done. But I do think it affected how many romaine lettuce heads were actually positive. It would have been very interesting to see what would happen if we had done this on day 28. So just some conclusions and some final thoughts here. We saw that the non-pathogenic E. coli, TVS 353, and the 0157H7 strains, they can survive in soils that were side dressed with organic fertilizers or biological soil amendments, and they could survive up to 4 weeks. But we did see that the level of E. coli transfer to romaine lettuce was low and sporadic. We saw higher levels of Salmonella infantis were found on romaine lettuce that were grown in soils side dressed with organic fertilizers, and that's compared to the unamended. And we did see actually more declines, faster declines, and lower survival of the Salmonella infantis and those that received no side dressing. And I guess the point I'd like to make here is similar to what Keith said, that maybe Salmonella and E. coli 7 are interacting differently with these soil amendments. Maybe there's specific interactions here that the Salmonella can maybe respond a little bit better, or utilize something, a nutrient, maybe find some adapted interaction with the microbial community. Maybe that elongates its survival a little more. It's hard to tell because our environmental conditions were so consistent in our growth chamber. We had consistent irrigation pattern, consistent, essentially, temperature, consistent relative humidity for the most part. And all those things don't happen in the field, right? These are things that happen in growth chambers. So maybe that consistency also affected the survival patterns that we see. We do see, for example, that rainfall makes a big difference, at least here in the mid-Atlantic region of the United States, when we do these field studies outside. So those are all things that I think could affect the survival patterns that we observe. But yeah, I think this is really interesting. I think we have more to investigate looking at some of these differences between E. coli 0157H7 and Salmonella. But what was good is that everything does decline. We didn't see any side dressing treatments or regimens that supported growth or survival at a steady state for these pathogens. With that, I would just like to thank all the people involved to help us get this research done. Our University of California, Davis collaborators as part of the contact team, our University of Delaware collaborator, Dr. Callie Neal, who helped get Ray on board here at USDA. Definitely our Objective 2 team, Keith and Laurel and Govind and Abhinav, but our leadership, Michelle Daniluk, who leads this project really well, Christine Kessler, who does everything behind the scenes, and Ari Huglar, who helped out with a little bit of the modeling expertise to get us to an answer. And then just our funding acknowledgments for the Contact Project and USDA ARS. And thank you all for joining us on a Thursday afternoon.
Wonderful. Manan, Keith, Laurel, thank you so much.